biological classification, the next kingdom is kingdom fungi. Kingdom fungi is a special group of individuals which are having the cell wall, but the cell wall is non-cellulosic in its nature, but at the same time they are eukaryotic in their nature. That is, they contains the true nucleus and heterotrophic organisms. That is, they cannot prepare their own food materials because they lack plastids in them, and cell wall is present. This type of organisms which are eukaryotic but non-photosynthetic in their nature. This group of individuals includes mushrooms, toadstools. Here all poisonous mushrooms are called as toadstools. So this toadstools includes Amanita species. Apart from the mushrooms, there are some delicacies which are having high nutritious in their nature. Morels and truffles are also been included in the fungi, and not only the nutritious one, but there are some organisms which are parasitic and some exist in the saprophytic. So this type of lot of different diversity is present in the group of individuals which are included in the fungi. Then these are mostly growing in the moist organic matter. That means wherever the organic matter is present, so where this moist condition is available, so in that moist atmospheric conditions they have high frequency of survival, and this fungi, the study of fungi is called as mycology. So actually the term mycology was derived from the myco means mushrooms, logos means study, the study of mushrooms, and mushrooms are the part of fungi. That's the reason why the study of fungi is also being known as a mycology. And as we said that some of them are parasitic in their nature, like albugo is one species which will form the special structures in the inner part of the leaves of some particular species of members, like in Solanaceae. So likewise they produce parasitic, they exhibit parasitic type of nutrition also. When we are talking about the saprophytic nature, so some of them are growing on the dead and decaying organic matter, so that is they are growing on the cow dung also, that is coprophilous, cow dung, and some of them are lignophilous. Lignophilous means which are growing on the rocks. So likewise a lot of diversity can be seen in the survival of this group of individuals. And in other systems of classification, fungi have been grouped along with algae, but the difference is algae are autotrophic in their nature, but fungi are heterotrophic in their nature. And the organization is called as a thallus. The body or body organization is called as thallus, and this thallus is either may be made up of unicellular or sometimes it may be made up of multicellular structures also. If they are multicellular, they may be sometimes filamentous. And here we can see the close interwoven structures are called as hypha or hyphae. So these are thin, fine, transparent, thread-like or filamentous structures by which the body of this fungal members is made up of, and many hypha are interwoven with each other to form the body is called as a mycelium. So here the interwoven structure of or interwoven structure made up of hyphae is called as a mycelium. So specifically the thallus of the fungus members is called as mycelium. So here the interwoven structure is called as plectenchyma. This plectenchyma will exist in two forms. One is called as prosenchyma and other is pseudoparenchyma. In prosenchyma, many the hyphal structures are arranged parallel to each other likewise. This is one hyphal structure, and one other hyphal structure is present parallel to this one, and one other like this parallel structures are arranged. This type of parallel arrangement of the hypha is called as a prosenchyma arrangement, whereas here the compact arrangement of isodiametric cells is present. This type of arrangement is called as a pseudoparenchyma. So in the schematic representation, so we can see this type of isodiametric structures. This type of organization where compact arrangement of isodiametric cells is present, this is called as pseudoparenchyma. That is the mycelium which is in the interwoven structure called as plectenchyma, which may exist in the form of prosenchyma or in the pseudoparenchyma manner. Then if you see the cell structure, here each cell, as we said that they may be either unicellular or multicellular, but if you consider each cell, it is made up of the cell surrounded by cell wall. This cell wall is made up of a special type of polysaccharide called as chitin. So this chitin is nothing, is a special type of polysaccharide which contains nitrogen. So we can call it as nitrogen containing polysaccharide molecule. So inside the cell wall, plasma membrane is present. This plasma membrane is similar to that of other organisms like which is made up of lipids and proteins, that is lipoproteinaceous. And inside this structure, inside the plasma membrane, we can see the presence of protoplasm, the fluid content called as protoplasm, which contains both the cytoplasm along with the nucleus. And if you observe the structure of the one of these examples, so this type of organization can be seen. So where septa may be present like this, and each cell will contains vacuoles like this. And along this one, we can also see the presence of nucleus, and this nucleus is a true nucleus or the definite nucleus which contains nuclear membrane, nucleoplasm, chromatin and nucleolus. And apart from that one, the cytoplasmic contents are present like this, and this is a plasma membrane. So outermost one is called as cell wall. The next one inside this one is called as plasma membrane, and here all cell organelles are present except plastids. That means they contains mitochondria, Golgi complex, endoplasmic reticulum and ATS type of ribosomes, but they lack plastids. So because of this character, so these are considered to be the heterotrophic in their nature. So here what's the basic difference that we find between the fungus group of organisms and other eukaryotic organisms is all other eukaryotic organisms contains plastids in the plant groups, not in animal groups, but fungus is a plant group, but it is not having the plastids despite of eukaryotic in its nature. Here if you observe the cell organization, the cells either we say that they may be unicellular or multicellular in their nature. Based on that nature, we can say that the septa may or may not be present. If septa is absent, we call it as aseptate condition, and if septa are present, we call that hyphae as a septate hyphae. And there is a deviation in the septate condition because in some of them the septa is non-perforated, that is non-porous. There is no perforation or connecting structures between the adjacent cells. This type of septa is called as solid septa. But in members of higher fungal group like in Basidiomycetes, so their special type of septa are present. Those septa are called as dolichopore septa. This type of structure is formed by some barrel shaped structure. This type of cap like structure will be formed around the barrel shaped structure. This type of pore, or we can say the pore is present. The pore is surrounded by the barrel shaped borders. So this type of septa is called as dolichopore septa. This is a special type of septa that can be seen in the higher fungal group of organisms. Here the reserve food materials are present in the form of glycogen. Then if you observe the nutrition, they exhibit heterotrophic type of nutrition in which some may be showing parasitic, some may be showing saprophytic, and some are exhibiting symbiotic type of nutrition. So in the symbiotic type of nutrition, the members of Ascomycetes and Basidiomycetes, they are associated with the members of algae, and that mutual beneficial association between an algal member and a fungal member is called as lichens. So lichen is one of the example of symbiotic association or symbiotic type of nutrition exhibited by members of fungi. And coming to the next one, that is reproduction. So these members of fungi will exhibit the three types of reproductions. One is vegetative, asexual and sexual methods of reproductions. Starting with the vegetative type of reproduction, here different methods of vegetative reproduction is exhibited by these members. Starting with the first one, the fragmentation. Fragmentation is the process where the filamentous structure, if it is there, that will be broken into smaller pieces. Like for example, if this is a filamentous structure that is present in the one of the member of fungus, so that may be broken into small pieces. Whenever they are broken, each part or each piece that have been separated, or the fragment which have been separated, that will develop into a new individual. So that will be similar to that of the parent. So likewise here the single parent will be participating in the fragmentation process. The next one is budding process. Budding can be seen in examples like in yeast, where the parent will form a small protuberance. So by the meantime, the nucleus present in this parent will divide to form two daughter nuclei, and one of the nuclei will enter into the protuberance. So this protuberance is called as a bud, and here one bud may be formed. Sometimes a chain of buds or multiple buds may be formed. Each of the bud will get detached from the parent and that will grow into a individual organism. So this is a way of vested reproduction where the budding help in the perpetuation of the progeny. Next one is binary fission. So many a times till now we had discussed about binary fission, that is if a unicellular organism is present, that will divide by cell division to form into two daughter cells, and each of that cell that is formed as a result of cell division, that will grow and act as a daughter individual. So this type of mechanism where nearly similar type of two daughter organisms are formed from the parent. Next one, sclerotia. So this is a structure which is formed during unfavorable conditions, or in order to tide over these unfavorable conditions. Here the compact group of hypha are surrounded by a membranous structure. The compact group of hyphae is covered by a thick covering during or to tide over the unfavorable conditions. This type of sclerotia can be seen in Penicillium example. So these are different methods of vested reproduction which occurs in different members of fungi. The second method of reproduction is the asexual method of reproduction. Here also the single parent will participate, and they form some special structures. Those special structures are called as spores, and these spores may be either uninucleate, sometimes they may be multinucleate also, and the spores may be either sometimes thin or thick walled, and in some of them they may be formed inside the sporangium, that is endospores, or sometimes they may be formed outside the structure, so exospores. And these spores are dispersed by different media, particularly by the wind, and they fall on suitable substratum and germinate to give rise to the new individual. So this type of special structures which have been formed are called as spores, and here the spores are of different types, starting with the first one, zoospores. Zoospores are nothing but the motile spores are called as zoospores. That means they have the special structures which are concerned with the locomotion, either it may be a cilia or flagella. So with the help of that locomotive structures, they will show the movement. The type of structures are called as zoospores, and these zoospores are formed inside a special structure called as zoosporangium. The zoospores are formed inside the zoosporangium. Example is saprolegnia. So next type is sporangiospores. Sporangiospores are formed in examples like in Rhizopus, where the sporangiophore is present. The sporangiophore will produce an spherical shaped structure called as sporangium, and the sporangium will have the haploid nuclei, and each of the nuclei will form some cell wall around it and converted into the spores. And each of this spore which is formed inside the sporangium, that's why we call them as sporangiospores. And these sporangiospores are generally haploid in their nature, and they have thin wall around them. Once the sporangium is broken or bursted, So all the spores are exposed to the outer environment, and with the help of the wind they are dispersed, and when they fall on suitable substratum they germinate and give rise to new individuals. So this type of structures which are formed inside the sporangium, so we call them as endospores. Zoospores are also formed inside zoosporangium. That means both the zoospores and sporangiospores, both of them are considered to be the endospores. Next, conidiospores. Conidiospores are the structures, or we can call them as conidia, which are formed outside like this. This is a cellular structure. So this type of structures are called as a conidia or the conidiospores. The conidiospores can be seen in penicillium, and these conidiospores are formed outside the structure, so we call these are considered to be the exospores. Then chlamydospores. These chlamydospores are formed during unfavorable conditions. So this is a structure which is formed by the accumulation of the cytoplasmic content in between the cells. So this type of structure where the thick walled organization is formed. This thick walled structure which contains the nucleus, haploid nucleus in them, these are called as a chlamydospores, and these chlamydospores are considered to be the resting spores because they are formed during the unfavorable conditions. That's the reason why they will be in the resting condition till they regain the favorable conditions. Once the favorable conditions are regained, so what happens, they germinate and give rise to the new individuals, and each of the chlamydospore may contains more than one nuclei. So those are multinucleated in their condition. Next one, arthrospores. These are called oidea. So these are formed when excess water and sugar substances are formed. So here what happens is they will segregate, or they undergo separation. When they undergo separation, each of that structure is called as arthrospore or oodia, and they will germinate and give rise to the new individuals. This type of structures can be seen in examples like in Rhizopus. Chlamydospores can also be seen in Rhizopus example. So these are different types of asexual structures which have been formed in various groups of fungal individuals to perpetuate and continue the race of the species. The third type of reproduction is the sexual reproduction. Here in this process where different mechanisms are operated, but basically we can say that three steps are involved in the sexual reproductive process. First one, the plasmogamy. The second step is called as karyogamy, and the third step is called as meiosis. So these are the three steps which are involved in the entire process of sexual reproduction in the fungal members. So plasmogamy, actually when the reproductive structures have been fused, directly the nucleus will not fuse, but what happens, only the cytoplasmic contents will fuse. The fusion of the protoplasmic or the cytoplasmic contents in which the nucleus remain separate. This type of first step is called as a plasmogamy, and after some time the individual nuclei which are present in that fused structure, they start fusing. That is called as a fusion of nucleus, called as karyogamy. Once the nucleus will undergo fusion, that will divide by meiotic division and leading to the haploid structures. So that is what the third step called as meiosis. So total three steps are involved in a sequential order, starting from plasmogamy, karyogamy, ending with meiotic process. In the process of sexual reproduction, we have seen that the fusion of male and female gamete, that is antheridium and oogonium, resulting in the zygote. But here, out of all the members of fungi, Deuteromystis, they lack sexual reproduction in the life cycle, whereas other group of fungi will exhibit sexual reproduction where the male and female gametes are formed. So the male gamete, male structure is called as antheridium and the female structure is called as oogonium. The fusion of this male and the female resulting into the zygotic structure which is diploid in its nature. And here there are different methods of sexual reproductions which are taking place in the fungal members. So the first one is the planogametic copulation. It is a process of fusion of, or we can say the fusion between two flagellated gametes. This type of fusion is called as a planogametic copulation. The second one is called as gametangial contact. Here in the gametangial contact, the two types of reproductive structures are present. One is called as antheridium, that is male structure, and the female structure is called as oogonium. The contact will be there between the two structures, and here the male structures will release a male gamete. This male gamete will enter into the oogonium, and it will fuse with the egg cell which is present inside the oogonium to form into zygote. This type of process is called as gametangial contact. The third one is gametangial copulation. In this, two gametangia are present. One is considered to be the, in some of them it is considered as a plus strain gametangia and other as a minus strain gametangia. So depending upon heterothallic or homothallic species. If it is a heterothallic species, plus strain and minus strain gametangia are present. Always the fusion will occur between one plus strain and one minus strain gametangia only, and never between two plus strain and two minus strain gametangia. And if it is homothallic species, so here the fusion occurs between two gametangia where there is no differentiation in their sexual behavior. So whatever may be that the two gametangia present which may contact, and the nucleus present in them will fuse with each other to form into the zygospore. Actually, zygospore is a structure in which the zygote is surrounded by a thick wall around it. So this type of structure is called as a zygospore which is diploid in condition. The zygospore will undergo meiosis to produce the haploid structures from where the new individual will be produced, which is haploid plant body. The fourth one is the somatogamous copulation. In the somatogamous copulation, the plus strained mycelium and the minus strained mycelium, or plus strain hypha and minus strain hypha are present. These two hypha will come in contact with each other. First, the cytoplasmic contents will fuse. That is the first one. Plasmogamy will occur, whereas the two nuclei which are coming from one plus strain hypha and one minus strain hypha will remain separate, and after some time both of them will fuse. In the mean process of fusion, before fusion, we can say that the structure will be formed where two nuclei present in the same cell. So this type of structure where two nuclei are present in the same cell, we call it as dikaryotic cell, and the hypha which contains this type of organization is called as dikaryotic hypha. Here one thing we have to note, we have to consider that the hypha which are fusing are called as monokaryotic, that is plus strain hypha is monokaryotic and minus strain hypha is also monokaryotic. Monokaryotic means each cell of the hypha will contain only one haploid nucleus. When the primary hypha, that is a plus strain and minus strain hypha, will fuse, it is converted into dikaryotic phase, where each cell will contain two nuclei. One is coming from plus strain, that is plus strain nucleus. One is coming from minus strain, that is minus strain nuclei. And this is a condition where the entire structure of that mycelium will contain dikaryotic cells. And after some time, the nuclei present in the dikaryote, each dikaryotic cell will undergo fusion, that is called as a karyogamy. And once the karyogamy occurs, single diploid nucleus is present in each of the cell. Now the cells or the nuclei will undergo meiosis. So when they undergo meiosis, they form some special structures called as, so in the meantime, before this structure, we can say that in ascomycetes they form special structures called as ascus, and in basidiomycetes they form special structures called as basidia. And the ascus, inside the ascus, the meiosis will occur to produce haploid structures called as ascospores. So if you observe the structure of ascus, it will be nearly club shaped with haploid structures inside them, and these haploid structures are called as ascospores, and this one is called as ascus. Whereas if we consider structure of basidia in the basidiomycetes, so the nucleus present in the basidia will undergo meiosis to produce 4 haploid structures. So this is a basidia or basidium, and they will produce 4 haploid structures outside. Each of the structure is called as basidiospore. So this type of structures can be seen, and each of the ascus will disperse and form into the new organism, whereas here in basidiospores also they segregate, they will separate and disperse to form into the new individuals. That is the way how these new structures are formed by the process of sexual reproduction in different groups of fungal organisms. Under kingdom fungi, there are 4 groups are there. Those are called as Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. So let us start with the first group of fungi, that is Phycomycetes. Phycomycetes group of fungi are called as algal fungi. These are mostly aquatic in their habitat, and they are growing in the moist conditions on the decaying woods, and on the moist habitats. They may survive as parasites on the plants. So this group of individuals are called as Phycomycetes, and these Phycomycetes members generally participate in asexual reproduction and in the sexual method of reproductions. In the asexual process, they produce the spores. There are 2 types of spores which are formed. One is called as zoospores, considered as a motile one, and the second type are called as a aplanospores. Aplanospores are considered as a non-motile spores, and the zoospores are formed inside the zoosporangium. The sexual reproduction This sexual reproduction may occur by formation of gametes. It may be either by isogamous condition, anisogamous, or oogamous type. Isogamous means the gametes which are participating in the fertilization are structurally and functionally similar, or we can say the fusion between structurally and functionally similar gametes. Anisogamy, here also the two gametes are participating. Here the fusion between structurally and functionally dissimilar gametes. Whereas oogamous condition is considered to be the advanced one, in which the male gametes are comparatively smaller and motile, whereas the female gametes are comparatively larger in size. They may be either non-motile, sometimes they may be motile also. So based on the type of or based on the nature of the gametes which are participating in the fusion, so three types of sexual reproduction may be possible in the Phycomycetes group of individuals. Then if you observe the structure of these members, they exhibit the presence of hypha. So these hypha are generally aseptate, and they have some sort of filamentous like structure, and as they are aseptate, when the reproduction is taking place, the nucleus will undergo free nuclear division to produce multinucleated structure, or cenocytic condition will be present in the hyphal structure of Phycomycetes members. Let us see the examples of Phycomycetes, like albugo. Albugo is a parasite on mustard plant, and other examples like mucor and rhizopus. So rhizopus is the one which will grow on the stale bread, and which is considered to be the bread mold, pin mold, or black mold. Sometimes it is considered as a weed of the laboratory. So here why it is so called as bread mold means it will grow on the stale bread. Pin mold means the rhizopus will produce asexual structures called as sporangiophore and sporangiosporangium. The sporangiophore and sporangium, both the structures appears to be like a pin and the pinhead. So that's why these are called as pin mold. And the black mold, so the spores which are formed inside the sporangium are darkly stained and black in color. So we can observe on the stale bread here and there some dotted structures which are black in color, so we call it as a black mold. And rhizopus will grow in the laboratory in the culture medium, spoiling the culture medium. That's why we call it as a weed of the laboratory. So these are some of the characteristic features related to the Phycomycetes members of kingdom fungi. In the kingdom fungi, the second group is called as Ascomycetes. Ascomycetes members are called as sac fungi, and this Ascomycetes members includes both unicellular, example is yeast, and some of them are multicellular also, like in penicillium. So here if you see the habitat, so or nutrition, So these are mostly growing in the areas where moist conditions are present. Apart from that one, these are growing as saprophytes, that is growing on dead and decaying organic matter. Some will be growing as coprophilous also. Coprophilous means which are growing on the cow dung, and some are growing as parasites, causing some damage to the other living organisms. Then these Ascomycetes members, if you see the cell structure, these are having two types of structures, primary mycelium and the secondary mycelium. And in that also, if you see the organization, so the hypha will be generally septate, filamentous and branched. Like this is a hypha or the mycelium, it is filament like, and it is divided into the small structures called as a branches, like this. And we can see the septa, which is dividing the entire hypha into individual cells. That is it is generally many cells are present, and each cell will contains a nucleus like this. So each cell is uninucleate in condition, and this vegetative structure is called as primary mycelium. This primary mycelium is monokaryotic in its nature. Monokaryotic means each cell contains single nucleus in it. But in the process of reproduction, where the two cells of the plus strain and minus strain hypha will fuse to produce secondary mycelium, this secondary mycelium is the structure which contains dikaryotic condition. We call it as dikaryotic hypha. That means when two hypha are participating, each structure will have many cells, and each cell will contain two nuclei. One nucleus from the plus strain, one nucleus from the minus strain cell. So this type of condition is called as dikaryotic phase. When dikaryotic phase is formed, after some time the two haploid nuclei, one is from plus and one is from minus, they will fuse to form diploid nucleus, and that process is called as dikaryo phase. Then this dikaryo phase, which is having two nuclei, will converted into a diploid cells containing structure. Then that is leading to a formation of the ascus, and this ascus is formed in a special structures called as fruiting body. The fruiting body in the Ascomycetes is called as ascocarp, and this ascocarp contains the ascus or asci. This asci, each of the asci will undergo meiosis. The meiosis will occur inside the ascus and produces haploid structures called as ascospores. So this is a way how this process is taking place, and here in the members of Ascomycetes there are three different types of ascocarps are present. Out of the three types, the first one is Cleistothecium. Here this ascocarp is closed type without any opening, and inside this Cleistothecium the ascus are present. So if you observe the structure, nearly spherical in its nature and surrounded by a wall, cleistothecial wall, ascocarp wall is present, and inside this one, so this is the other the body of that mycelium. Here inside which we can see the ascus, or plural we call them as asci. Along with the asci we can see some sterile hair structures called as paraphysis also, and each of the ascus will contain ascospores. And meiosis will occur inside the ascus, resulting into haploid ascospores. So this is the diagram of cleistothecium. The second one is perithecium. In perithecium, it is having a apical opening and the shape of this one is flask shaped. So here this is a structure of perithecium in which the apical opening is present. It is surrounded by this wall-like structure, and here we can see some hair-like structures which are present inside this opening. Inner wall of this one contains the ascus. Along with the ascus we can see the sterile hairs called as paraphysis, and here also the same thing. Meiosis will occur in the ascus, resulting into haploid ascospores. So this type of structure is called as perithecium. Then the third type is called as apothecium. Apothecium is a fruiting body of Ascomycetes in which it is saucer shaped. Here this is the organization where the mycelium will be present. The secondary mycelium, dikaryotic mycelium will be present here on the lower side, and in which the ascus will be present. And ascus is intervened by the sterile structures called as paraphysis. So these are the ascus, and inside which the ascospores are present. So like these different types of fruiting bodies are produced by different members of Ascomycetes. Ascomycetes members will participate in the asexual reproduction also. That is, they produce conidiophore, and the conidiophore will produce the conidia. These conidia are haploid structures which are produced outside. So these are exospores. So this type of asexual reproduction is present in different members of Ascomycetes. Some of the examples of Ascomycetes members are Aspergillus, Penicillium, Claviceps, Neurospora. Neurospora have significance because it is most widely used plant for the study of genetics, or it is very much used in the genetical studies. And other group which includes this one is morels and truffles. These morels and truffles are known for their delicacies, highly nutritious. So these are some of the examples of Ascomycetes members. Under kingdom fungi, the next group are Basidiomycetes. Basidiomycetes includes mushrooms, bracket fungi, and puffballs. These members they may grow as saprophytes, which are growing on the dead and decaying organic matter, and some of them are growing as parasites also. So parasites like Ustilago and Puccinia. Ustilago, which causes smut disease, and Puccinia causes rust disease. So likewise some of them are growing as parasites and some of them are growing as saprophytes also. Then if you observe the mycelial structure, here the mycelium is septate, branched and filamentous structure. So here one special feature is there which is nearly similar to that of Ascomystis, that is it will have primary mycelium which is monokaryotic and secondary mycelium which is dikaryotic. The primary mycelium will participate in the fusion of the cells, which leads to the formation of plasmogamy resulting in the dikaryotic cells, and that hypha which contains dikaryotic cells is called as dikaryotic hypha or secondary mycelium, and the nucleus after some time fuse to form into diploid cells, and the diploid structures will lead to the formation of the fruiting bodies. Here the fruiting body is called as a basidiocarp. Here this basidiocarp contains basidia. Inside this basidia meiosis takes place, and the basidia, once the meiosis will occur, will produce 4 haploid structures. So this is the basidia formed on each of the basidiocarp, leading to the formation of 4 haploid structures as a result of meiosis. These are called as basidiospores. So these basidiospores, when they germinate, once again they will give rise to the primary mycelium of the plant body. This is about the sexual reproduction which occurs in the members of Basidiomystis. Generally, asexual reproduction is not found in these members, and the vegetative reproduction is present, which occurs by a process called as fragmentation process. Likewise, these are different methods of reproductions which occur in the members of Basidiomystis. Let us see some of the examples of Basidiomystis. So, as we said that some of them are mushrooms. The best example of mushroom is Agaricus bisporus. So, Agaricus bisporus will produce the mushrooms which are called as white button mushrooms. Are European mushrooms, and these mushrooms are the cultivable species. And one more thing, that mushrooms contains both the edible and also the poisonous mushrooms. We cannot differentiate the edible mushrooms from poisonous mushrooms based on their morphology, and edible mushrooms are generally cultivable species which are having highly nutritious in their nature, which contains a protein content and more than that of the non-vegetarian and the vegetarian foodstuff that is consumed by human beings. And apart from that one, the mushrooms are considered to be the most ideal food for the patients who are suffering from diabetes and hypertension. Why? Because they lack or very little amount of fats, and fats are present in them. Carbohydrates are very less, starch is also very less, and apart from that one, they contains minerals, vitamins and organic substances which are required for a healthy growth of human beings. Then the poisonous mushrooms are also there, which belongs to species called as Amanita species. At the same time, Ustilago is one species which causes smut disease, which is a parasite, and Puccinia is the one which causes rust disease. And bracket fungi is polyporous. Puffballs example is Lycoperdon. So likewise, these are the different members of Basidiomycetes which have their own significance in terms of the morphology and physiology. The last group of fungal kingdom are Deuteromystes. So Deuteromystes members are called as fungi imperfecti or imperfect fungi. These are so called as imperfect because they lack sexual stage. They are represented only by the asexual stage, and this is the only group where the sexual reproduction is absent. Next, if you observe the organization of the thallus or the plant body, the hypha is a septate and branched filamentous structure as like that of other fungi. Here the special feature related to Deuteromystis members is the asexual stage is given one name and the sexual stage is represented by some other name, and sexual stage is present in some other group, whereas asexual stages are kept in the Deuteromystis. Once the scientists find the relationship between the asexual stage present in Deuteromystis to that of any sexual stage, then the asexual stage which is present in Deuteromystis will be removed, and it have been given a certain specific place based on whether it belongs to Ascomystis group of individuals or Basidiomystis group of individuals. The sexual stage related to this Deuteromystis members, either they may belongs to Ascomystis group or Basidiomycetes group. So once they correlate the characters to the Ascomystis group, so the member which is present in Deuteromystis will be given certain position in the Ascomystis by removing it from the Deuteromystis. In the same way, if the characters of a sexual stage are more similar to Basidiomystis, so they have been given specific position in the Basidiomystis by removing them from the Deuteromystis group. Likewise, the special characteristic features related to this Deuteromystis are existing, and these Deuteromystis members will in the form of saprophytes or in the form of parasites. So as we know that if they depend upon dead and decaying organic materials, we call them as saprophytes, and if they are depending upon living hosts, we call them as a parasitic organisms. So here when they are the parasites, they cause different diseases in the host on which they are surviving, like Alternaria, which will cause early blight disease in potato, and other example is Colatotrichum, which cause red rot disease in sugarcane. And when they are present in the form of saprophytes, they are considered to be the decomposers, and when they exist in decomposers, they are useful for recycling of minerals. So this is one other functional significance related to the Deuteromystis members. These are the four groups of fungal members which are kept under kingdom fungi.